Hello everyone, and welcome to our online lecture series for Bio 2. Today we finally move on from our first topic where we visited a lot of different ideas in terms of transmission genetics, how things called genes and their versions called alleles get transmitted through the process of meiosis through their progeny, and how different interactions between genes calls for different modes of inheritance of different traits. Today, and for our next shorter section, we're going to be exploring the actual structure, function, and end products of DNA. The entire topic is wrapped up in what's called the central dogma of biology. The belief that one gene will code for one protein, and that this tenet of one gene, one protein is critical to our understanding of how biology and DNA work. So, we first learned about the existence of DNA and isolated DNA in the 1860s. This individual here, Friedrich Miescher, was the first biologist to extract DNA from a nucleus. He did so in one of the labs of this beautiful place called Schloss Hohentubingen, which literally means fancy house or castle on the hill in Tübingen. And Tübingen is coincidentally where I did my postdoctoral work, so it was really cool to get to walk in the same building where DNA was first isolated. But we'd have to wait quite some time afterwards to actually get some idea about what the nature of DNA was. Miescher just extracted it and showed that it was acidic. It would take the experiments of Griffiths, Hershey Chase, Chargaff, and Watson and Crick with Rosalind Franklin in order to fully elucidate the role of DNA. So let's go into those experiments briefly. The first of these investigations were called the Griffiths experiments. And Griffiths was studying two different strains of a virus, the R strain and the S strain. The R strain was non-lethal. The S strain was lethal. And Griffiths would expose mice to these different viral strains. Now, when he gave a mouse the R strain, it would live. When he gave a mouse the S strain, it would die. He then tried to kill the S strain, the lethal strain, with heat and injected the mouse with the virus and found that the mouse lived. However, when he gave the S strain that was heat killed plus the R strain in one solution, so mixed together, meaning the dead pieces of the S strain and the living R strain, the mouse died. And he found that there could therefore be some component of the dead bacteria that would confer, or not bacteria, but virus rather, that could confer the transmission of lethality to the mouse. So, giving scientists an idea that there had to be something in this virus that could pass from one dead virus to the living virus and still confer that lethality. These experiments were done in the late 1920s, 1928, and then there were follow-ups in the 1930s and 40s, most notably the Hershey Chase labeling experiments. And these kind of really established that DNA was the hereditary information, and they received the Nobel Prize for their work. The gist of their work was showing what was a two-step process. So first, they prepared viruses that either had radioactively labeled DNA or radioactively labeled proteins. They then infected a bacteria with that virus. The information, or rather the DNA, was inserted into the bacteria, as we'd later learn. Step 3 was that the cultures were blended and centrifuged to separate bacteria and virus. And then step 4 was seeing what was left over in the culture. Well, when the virus was replicating inside of the bacteria, with a protein coat label, there was no radioactivity labeled in the bacteria. With the DNA labeled, there was radioactivity left in that bacteria. This showed that the piece inserted was actually the DNA, not the protein. And from this, they were able to conclude that it is DNA, not protein, that is the hereditary information of life. Pretty clever, I think, right? Next up, in order to elucidate the structure of DNA, we had some experiments performed by Chargaff. And Chargaff's experiments were kind of looking at the composition of DNA across different organisms. And what Chargaff noticed was that there were 4 different bases in DNA. He gave those nucleotide bases names: cytosine, thymine, adenine, and guanine. And Chargaff noticed that the amount of cytosine, C, was always equivalent to the amount of guanine, G. He also noticed that the amount of adenine, A, was equivalent to the amount of thymine, T, regardless of whatever organism he looked at. If he isolated DNA from a plant, this was true. If he isolated DNA from a human, this was true. If he isolated DNA from a dog, this was true. This allowed Chargaff to make the conclusion that cytosine paired with guanine and that adenine paired with thymine. Once we got to some organic chemistry, with a better understanding of the direct structure of these molecules, we were able to see that the adenine and the guanine have 2 rings in their nucleotides. We call those 2 purines, and you can keep track of which are the purines because purines are pure as gold. That little mnemonic of PAG, pure as gold, helps you remember that adenine and guanine are your purines. Cytosine and thymine, in contrast, only had a single ring to their nitrogenous nucleotide. We call those molecules pyrimidines. Now, it wasn't Chargaff's work, but it was noticed later on. I do want to include the RNA substitute, uracil, which takes the place of thymine, but only in RNA. You'll notice that by pairing a pyrimidine with a purine, how many rings do you get going sideways? Well, if I pair something that has 2 rings with something that has 1 ring, the width of that DNA molecule will be 3 rings. Similarly, a 1-ring pyrimidine with a 2-ring purine also makes 3 rings as to the width of a DNA molecule. So we'll be looking at this later, but just keep in mind that the width is always going to be 3 rings across when you're looking at the structure of a DNA molecule. Now, I don't need y'all to keep track of glycosidic bonds, but I do want y'all to know that the sugar backbone of DNA is made of ribose. the sugar backbone of RNA is made of ribose, and the sugar backbone of DNA is deoxyribose. So they have the same 5-carbon sugar backbone, but one has an extra oxygen compared to the other, with ribose having one more oxygen than deoxyribose. Does make sense, right? DNA. Sorry about that. The other components of our nucleotides, in addition to that sugar backbone and that nitrogenous base, with these being our nitrogenous bases, are phosphate molecules on the backbone. Those phosphate molecules help protect the back of the sugar, as that charge provides an electrostatic barrier. So those are 3 components of each monomer of our DNA, the basic building block of DNA. A phosphate backbone, a sugar ladder, either ribose or deoxyribose, and a nucleotide middle. But we still didn't know how all of this stuck together. How did it all look when it all became pieced together? To understand that, we had to wait for the revolutionary work of Rosalind Franklin, the female scientist who had the skill to actually take X-ray crystallography pictures of DNA, and this is what she saw, and then the 2 scientists, Watson and Crick, who would take a look at Rosalind's work, perhaps not with her permission, and deduce that DNA was actually a double helix. So they looked at that picture and put together all of the other work from Chargaff and colleagues to piece together the components of DNA to finally put together that double helix model that you all recognize in this image here. A sugar phosphate backbone with nitrogenous base pairs in the middle, those nitrogenous base pairs always being 3 rings across. With thymine pairing with adenine, I will draw attention to the fact that the thymine-adenine pair has 2 hydrogen bonds, whereas the guanine-cytosine pair has 3 hydrogen bonds. This makes the GC bond stronger than the AT bond. Something to keep track of. And then, of course, we have some information about the major and minor grooves of DNA, but that's mostly beyond the scope of this class. Okay, so that was a lot of information thrown at you, probably quite quickly. So let's make sure that we understand the major takeaways from these DNA experiments so that we can talk about the fundamental process of DNA replication afterwards. So what do we know about DNA right now? Well, we know that DNA is the hereditary information of life. Oops. This is in contrast to folks who thought that it was actually protein that was that hereditary information. Those experiments conducted by Griffiths and Hershey Chase went on to prove that. We also know that DNA is made up of a phosphate protective backbone, and it's this phosphate backbone that gives DNA a negative charge. It's this charged feature that allowed scientists like Miescher to isolate DNA in the first place. In addition to the phosphate protective backbone, we have the sugar ladder that makes up DNA and RNA, with DNA being made up by deoxyribose and RNA made by ribose, but in both cases, they are a 5-carbon sugar making up that ladder. Now, in between these ladders and the protective backbone, you have nucleotides, these nitrogenous bases. The nitrogenous bases are either pyrimidines, like cytosine and thymine, or purines, like adenine and guanine. Purines, pure as gold, have 2 rings in their structure, whereas pyrimidines have 1 ring. And you are always going to pair a pyrimidine with a purine, adenine with thymine, and guanine with cytosine to get a 3-ring width to your DNA molecule, leaving you with a structure that looks double helixed overall and has different amounts of hydrogen bonds, with GC having 3 of them and AT having 2 of them. All right, so there's our fact sheet about DNA. With this information, we can now embark on the real take-home message of today's lecture. Not that you don't need to know this, though, of course, but with this, we can finally get to our biological model of DNA replication. Because once we understood how DNA was built, we wanted to understand how it gets copied. Because this is the thing that has the information for life. It is necessary to keep us functioning. So how do we go about making enough copies of it without destroying or changing it? We know that DNA mutations give rise to all kinds of different functional consequences, like our alleles, right? Whenever you get a mutation, you have a new allele of a gene. So how do we keep most of our genes from having those mutation errors? And how in the first place do we actually end up with the same information held in our double helix DNA molecule? So we're first going to take a look at a model from Wikipedia, and then we're going to draw it out ourselves, going step by step over what happens in order of how it occurs. So, the first thing to note about the process of DNA replication, and the first vocab term associated with this, is that it is a semi-conservative process. What this means is that you will start out with 2 DNA strands. And in order to copy them, you will end up with one of the original strands in each of the new pieces. From these original strands, we are able to make an identical copy of what used to be there attached to it. And this is what semi-conservative means. Rather than having one piece that was completely the old and having it just generate an entirely new double red banded copy, you instead distribute those old pieces of DNA across each of your 2 new pieces of DNA. Hence, semi-conservative. In terms of structure, and I don't want you to be overwhelmed by this, we will be going over this a couple times. there are a lot of different components to the replication of DNA. So let's take a look at them in turn. The first thing you're going to note is that your DNA has directionality. It has a 3' end and a 5' end. Now, what the 3' and 5' refer to are actually locations on your 5-carbon sugar, but unless you are familiar with organic chemistry, we don't need to keep track of that. All we need to know is that there is a 3' end and a 5' end. And I want you to keep track of the fact that DNA will always build, starting at the 5' end and build towards the 3' end. This is called the leading strand of DNA. And the reason why it is leading is because the enzyme that copies DNA is able to just walk along in one direction on the leading strand up until the job is done. When you are trying to copy the other side, meaning going from 3 to 5', you instead, because of this enzyme, can only go in the direction of 5' to the 3'. So you're going to have to build in chunks. If I'm trying to build something from 3' to 5', but I can only build 5' to 3', what that means is that I'm going to have to build small pieces and then assemble them together. This leads us to our leading strand and to our lagging strand. The side that's assembled in pieces is our lagging strand because it takes multiple runs of our copying enzymes in order to actually piece together the entire component. Here on the right, we also have all of the enzymes necessary to complete DNA replication. Topoisomerase is our enzyme that's going to be making sure that there are no tangles up front. And you see it here in the Wikipedia drawing. Topoisomerase makes sure that your DNA does not get tied up in a knot before the unzipping happens. The molecule that does the unzipping is helicase. Helicase breaks down the helix, therefore we call it helicase. You'll notice its presence at the point where the DNA strand is being unzipped. Right after the helicase, we have naked DNA nucleotides. That's not very safe. We don't want them to be out in the world alone without something protecting and covering them. Those things protecting and covering them are our single-stranded binding proteins. Or SSBPs. So those SSBPs, seen in purple here, are keeping those nucleotides that are unzipped, but as of yet unbound to a new pair, safe. Our next enzymes are involved in the replication process. DNA primase is going to be the signaler for our start. So you'll notice that DNA primase is putting down an RNA primer so that way our DNA polymerase, in this case Pol 3, knows where to begin copying. So this is your start signal, and this is your copy base enzyme. Your DNA polymerase 3 is the molecule that can build in the 5' to 3' direction. That means that this is always going to be sliding in the direction of 5' to 3' and building in a leading fashion because of the activity of DNA Pol 3. After DNA Pol 3 has added the bases and copied the bases, you then have DNA Pol 1 replace the RNA primer. So you see that function happening back here. The lagging strand, because it's building these in much more sections, we can see that it requires a lot more RNA primers. Every single start location needs a primer. These different chunks that you're building it in were discovered by a scientist named Okazaki, and therefore we call them Okazaki fragments. Your DNA polymerase 1 is going to go to each of your Okazaki fragments and replace the RNA primer that was put there by primase with DNA. But it does not stitch everything together. These fragments are still separated, as you can see right here. In order to connect them, we have to ligate them, and that is the role of DNA ligase. So, DNA ligase will come in after DNA Pol 1 and connect our separated Okazaki fragments. Now, while DNA Pol 3 is going through and copying bases, we'll get to a discussion of how fast exactly that occurs. The enzyme that's making sure that DNA Pol 3 does not slip off is called the sliding clamp. And this is because Pol 3 can be a very, very fast enzyme. In fact, in prokaryotes, DNA Pol 3 can do something like 1,000 bases per second. It needs that sliding clamp to make sure it does not fall off at such blistering fast paces. It's a little bit slower in eukaryotes, but there are a lot of other components to the eukaryotic genome that make it necessary to be so. But at the very least, we now have an idea of what this semi-conservative process looks like. You're going to have your two different strands, 3' to 5' and 5' to 3', one being leading, one being lagging. In all cases, topoisomerase is going to come unwind the DNA to make sure there are no knots up front. Helicase is going to come unzip the DNA. Those single naked nucleotides are held safe by single-stranded binding proteins. Your primase is going to come to those single-stranded binding proteins and naked nucleotides to attach an RNA primer to tell DNA polymerase 3 where to start. DNA polymerase 3 will bind at that RNA primer and start copying the bases. It's kept firmly in place so that way it does not slide off the DNA by the sliding clamp. DNA polymerase 3 builds in a direction of 5' to 3'. Once DNA polymerase has added all of the nucleotides, We then have DNA polymerase 1 come through and replace the RNA primers. They're not stable, after all. They're RNA in a DNA molecule. Once the RNA is replaced, ligase will come in to connect all of our fragments. And last but not least, the DNA molecules will separate. So now you have that semi-conservative process whereby each of the old strands are split and incorporated in a 1-to-1 ratio in our new strands. Whew! So that's quite a bit. Let's clear the slate, just leave our vocabulary words up here, get rid of all the drawings and writings, and draw this process out together, because this is what will be expected of you come time of the midterm. So please grab a new piece of paper with me and draw along. So this will obviously be, oh, and we have a table here that summarizes the action of each of the enzymes, so if you want to, you can go ahead and pause it here. But I will continue on down to our blank slate where we've got kind of a shorthand of what each of the enzymes does. Topoisomerase preventing knots, helicase unzipping, SSBPs keeping those single nucleotides safe, primase giving that first RNA primer to start things, DNA polymerase 3 copying the bases, DNA polymerase 1 replacing the RNA primers, ligase connecting strands, and the sliding clamp keeping Pol 3 attached. We'll start by actually drawing our strands of DNA. So rather than trying to keep things complicated and swirly, I'm just going to draw DNA as single lines. So we'll have the unzipped portion of DNA and the zipped portion of DNA to the unzipped portion, the zipped portion to the unzipped portion. So you'll have connections here that still need to be undone by helicase. Now, in terms of directionality, we will label each side of our DNA strand with the 3' end up here, therefore the 5' end down here. And if this is the 3' side, that means that this has to be the 5' side, and this is the 3' side down here. Now, what does that mean in terms of leading and lagging? Well, if I have a 3' to a 5', that means the opposite side is going to be the 5' to the 3'. This is our leading strand. In contrast to that, each of our components here will have to be 3' to 5'. But you can only build 5' to 3'. So rather than zipping this way and building up, we know that the lagging strand has to be built in sections as things are unzipped backwards. To our enzymes, we have our first topoisomerase up front, making sure that things are not knotted. After our topoisomerase, in red, we will encounter our helicase that is doing the unzipping. Behind our helicase, we have our single-stranded binding proteins that are keeping the unzipped DNA safe. Then, in order here, we will have an RNA primer molecule attached to our start locations with primase. So, 1, 2, 3, draw some little stars indicating the RNA primer added by primase. Now, because you have to have multiple start sites here, you're going to have multiple RNA primers on the lagging strand. After our RNA primers are present, we can go to our DNA polymerase 3, because DNA polymerase giant green brick. We'll find where the primers are, attach to them, and slide and build. And behind it, it'll have all those bases added, and in front of it, it will have the single-stranded binding proteins. Now, in order to make sure that that clamp, or to make sure that that polymerase does not slide off, you have the sliding clamp placed on the DNA strand where the DNA polymerase is. So they're always working together. Your sliding clamp is always associated with your DNA polymerase. Now, in the same way that bases are being added to the leading strand, we also have bases being added to the lagging strand, but keep in mind that they are going backwards. Once the DNA polymerase builds this component, it'll then find another RNA primer here and build here, and so on and so forth. So it's kind of doing this leapfrogging strategy where it'll bind to where RNA primers are present, scoot down, build, reach where the end point is, then bump off, and come back to the next RNA primer. Build, bump off, come back to the next RNA primer. Build, bump off. So you've got multiple DNA polymerases, DNA polymerase 3 here, with its own sliding clamps. They travel as a package. Now, after your polymerase 3 has gone ahead and added bases, the RNA primer needs to be removed. So we have our DNA Pol 1 that's going to come in and replace those RNA primers with DNA molecules. So you're going to have more Pol 1 activity on the lagging compared to the leading strand. But then after things are connected, or rather after things are added, we then have DNA ligase, which is going to come in and stitch our two strands together. Whereas your DNA that is on the leading strand is just going to have one connection point where the break point was. Your DNA on the lagging strand is going to have a lot more connections to be made. So you see, hopefully, how all of this works in concert. The topoisomerase unwinding, the helicase unzipping, the single-stranded binding proteins keeping single nucleotides, open nucleotides safe, the RNA primers being added by primase, the DNA polymerase 3 sliding down the DNA molecule from a 3' to a 5', from a 5' to a 3' directionality, meaning that this side is our leading side, this side is our lagging side. Sliding clamps held on, helping hold our DNA polymerase 3 on target and without slipping. And then after polymerase 3 has come through and extended the DNA, we then see DNA polymerase 1 come by and replace those bases, and then ligase coming by to connect things, connect all of our disparate DNA molecules. So, broadly speaking, DNA replication happens in 4 different steps. Let's grab a little thinner marker here. Step 1 is our unwind step. That involves your topoisomerase, your helicase, and your SSBPs. Step 2 is our primer step. This is done by DNA primase. Step 3 is going to be our extension step, where DNA Pol 3 is adding bases. And then step 4 is our repair and connect. And in the repairing and connecting stage, you're going to have the DNA Pol 1, replacing RNA primers, and ligase connecting disconnected strands. Now, this entire unzipping process will always occur at what is called a replication fork. So while this leading lagging is happening this way, you also have the fact that this DNA is in a bubble, and therefore it's happening in the other direction with things flipped around. Hence why it's a fork. Two paths to that road. Now, the best way to study this, the best way to make sure that you have a good understanding of this, a functional understanding, so that way on the midterm you can explain this to me, is by drawing it out, making sure you understand all of the components, making sure you don't miss a step, making sure you understand the function of all of the enzymes. So please, pause the video now and try to draw this out again on your own. See if you're able to do it with your notes first, then without your notes after that. I'll take my time here. All right, so hopefully you've had some time. I took a little break there, paused the video, moved along, gathered my thoughts for our last little discussion of how we'll wrap this up. So we'll end today's lecture, acknowledging the fact that the most important thing that you need to know from this is this process of DNA replication, but then ending on things that we'll have to carry on into our future lectures. Most importantly, looking at differences between prokaryotic and eukaryotic replication, and then comments about DNA replication errors. So how do mutations occur? How do we mess this process up? Well, overall, as I mentioned before, there are thousands of nucleotides being added per second in prokaryotes and hundreds per second in eukaryotes. This extremely fast rate of incorporation actually leads to errors. On average, 1 in every 100,000 bases added is a misincorporation. It's an error by DNA polymerase III. There is a series of repair enzymes called the nucleotide excision repair complex that goes through and tries to identify these errors. After proofreading, the error rate ends up being much, much less. It's 1 in 100 million. So you add 3 zeros in order to get your proofreading error rate. But that still means that we'll be generating some amount of mutations every single generation. Mutation is a feature of DNA replication, not a flaw. I'll be saying that a lot. That mutation is a feature, not a flaw, of DNA replication. Some other differences when we're looking at prokaryotes versus eukaryotes, prokaryotes being those things without membrane-bound organelles and eukaryotes having membrane-bound organelles, meaning eukaryotes have a nucleus, prokaryotes don't. In terms of that origin of replication or fork of replication, prokaryotes have a single, whereas eukaryotes have lots and lots and lots of them. After all, eukaryotes frequently have bigger genomes, so in order to try to accomplish this process at a respectable time frame, eukaryotes have many origins of replication. Lots of different polymerase types, many more in eukaryotes than prokaryotes. So we talked about strand elongation being done by Pol 3, whose equivalent is some combination of Greek letters in eukaryotes, but I'll just be asking y'all to keep track of Pol 3, and then RNA primer removal by Pol 1. Some different enzymes doing these things in eukaryotes. And then our sliding clamp in prokaryotes has an analog called the pCNA. More on that in genetics if you opt to go the biology route. The other component that is very unique to eukaryotes is the presence of telomeres and a telomerase. So telomeres are present in linear chromosomes, which you and I have, and not present in circular chromosomes like prokaryotes have. So there has to be a special extension enzyme called telomerase that adds bases to the ends of chromosomes. Now, interestingly, telomere length has been correlated with longevity. It's one of the few things that if we sequence and figure out how long they are, it can be predictive, approximately, as to how long you are expected to live. Think about how crazy that is. It's kind of like scientific fortune telling. Not always, to the day accurate, not going to be that level of accuracy, but it does indicate overall longevity when taken on an aggregate across humans of different ages. And scientists are currently working on trying to figure out ways to extend telomeres to see if that also correlates with an extension of lifespan. The other thing that I'll want you to keep track of once we get into our biotechnology section at the end of this unit is what the error rate is in our own sequencing technologies. So when we do sequencing, meaning we try to figure out what is the order of nucleotides, ATGC, what is the order of those to make up the human genome? Well, we have a much, much higher error rate than the native error rate inside cells. Our error rates go from as good as 1 in 10,000 to as bad as 1 in 4. Ugh, that's really bad. We are constantly improving these. So it was 1 in 4 with a new technology called PacBio while I was in grad school. They've now got it under control to the point where they have an error of like 1 in 25 or something like that. So they're getting much better. They do have different advantages, and as you continue on in your career in science, you'll find out the advantages and disadvantages of each of these different sequencing technologies. But just giving you some background as to how DNA replication errors and differences between prokaryotes and eukaryotes tie into the DNA replication process. So we'll end there, but I will, of course, ask you again, one more time, after this lecture, without your notes, go over the process of DNA replication. Good luck, and have fun.